Thank you very much for tuning into our Young Women in Bio virtual presentation. My name is Edwina Kinchington, and I am the chair of the Young Women in Bio Pittsburgh. We're very excited to announce the availability of these online resources through our brand new YWIB online platform, which provides young women across the United States and Canada free access to our educational and leadership programs and other opportunities in science, technology, engineering, and math. YWIB is launching this new initiative for young women who are curious about STEM and who want to learn more but may not live near one of our 13 WIB chapters in cities across the United States and Montreal. Young Women in Bio gives girls today the inspiration and support they need to become tomorrow's leaders in STEM. We strive to provide education and hands-on experiences, as well as we share our passions in all fields that are scientific. The launch of YWIB Online comes at a time when there's a real need to provide young girls who cannot otherwise attend our events and programs with access to online programming. This will help our goal to ignite curiosity and to inspire young women to see a future for themselves in STEM. YWIB is part of the Women in Bio with chapters nationwide and in Canada. WIB is a nonprofit volunteer organization that promotes diversity and inclusion for women in the life sciences and beyond. YWIB partners with leading companies, universities, hospitals, and other organizations to hold engaging and educational programs for young girls interested in STEM, and young boys and young men too. In fact, in 1 year, we had more than 70 events across North America, reaching more than 4,500 young women and had the support of over 250 volunteers. With YWIB Online program, we will be able to reach even more young women. If you are interested in joining or helping or being a volunteer in one of the 13 chapters, please visit our WIB Women in Bio website, and that's listed here, and to learn more about YWIB also. Today, we're very fortunate to have two investigators from the University of Pittsburgh, Dr. Tullia Bruno and Dr. Andrea Gambotto, to share their journey of how they became successful scientists in the field of immunology and vaccines. Both investigators work at the University of Pittsburgh. Dr. Bruno is an assistant professor in the Department of Immunology at the University of Pittsburgh and a faculty member in the Cancer Immunology and Immunotherapy program at the Hillman Cancer Center. She obtained her Ph.D. from Johns Hopkins University in 2010 and completed her postdoctoral fellowship at the University of Colorado in 2015, both with a focus in tumor immunology. Dr. Bruno's research lab focuses on studying immunity with cancer patients, which makes her research highly translational with the potential for future clinical trials targeting cells of the immune system like B cells. In addition to her work in cancer, Dr. Bruno has been co-leading efforts at the University of Pittsburgh to study the immune response in local individuals that have been infected with COVID-19. Dr. Gambotto is an associate professor of surgery at Pitt. He received his medical degree in 1994 from Bari University School of Medicine in Italy. He followed with a postdoctoral fellowship at the University of Pittsburgh School of Medicine, studying gene therapy for heritable diseases. The passion of his work has been focused using adenoviral-based vectors to develop vaccines for Ebola, HIV, SARS, human papillomavirus, influenza, among others. In addition, he is very interested in using these novel antiviral therapies, adenoviral therapies for gene therapy applications. Thank you both for joining us today to talk about your journeys as scientists and about the topic of vaccines and immunity in this COVID era. As a reminder, please stay on mute during the presentation. We will take questions at about 4:40 p.m. You are welcome to submit questions in the chat as you think of them during the presentation. This presentation is being recorded and will be available at YWIB Online. Thank you very much, Drs. Bruno and Gambotto. At this time, Dr. Bruno, I will stop sharing and you are welcome to take over.
Great. Thank you so much for the invitation to be here today. I'm very excited, and I think Andrea and I's expertise are very complementary to each other in this subject. So I'm excited to talk with him on this subject matter. So let me go ahead and start the slideshow. Okay. So my goal today is really, as an immunologist, to talk about the immune system and the immune responses that we're seeing in our COVID studies. I won't go into a lot of detail on some of the work we're doing today, just because it's still developing. But what I hope to accomplish is to talk about why the immune system is important, particularly for all viral infections, and also in thinking about vaccine development, which I think will help set the stage for Andrea's talk. So the first thing I just want to mention on this slide is what you can see here is an image that we put together in my group. So a lot of what my group does is spatial imaging, where we look at cancer tissues and we look at the immune response in cancer tissues. And we can use this technology in any tissue. So what you're looking at in this background slide is actually a human tonsil, which is a normal human tonsil. And you can see quite clearly these really beautiful centers that are here. And they're actually called germinal centers. And I bring them up because I'm going to come back to them at the end, because they are the site where the immune system really gets educated against viral pathogens, against cancer antigens, etc. And I'll talk a little bit about what's happening in COVID patients and why some of what we're seeing in these patients has been a challenge. If we run out of time during the question and answer, just email me. I put my email address up here for your access. So before I go into that, I wanted to talk a little bit about my mentorship journey and what it means to be a principal investigator or PI in a lab. Just given the nature of this webinar, I think it's important to kind of talk about my trajectory. So basically, a principal investigator or a PI is a scientist. Scientist is our general term, but really, what I do is I run a research group at the Hillman Cancer Center. So a lot of the teaching that I do of either graduate students or medical students or even high school and college students, because we have students come to the lab and do research both through our summer program as well as undergraduates come from the Pitt campus. We really design experiments and try to understand what's happening in those experiments and teach by doing, basically. So I don't teach a lot of didactic classes or classes where I'm just teaching the material. I do that sometimes, but I'm really teaching hands-on in the lab. And to that end, we're really helping generate research ideas for experiments so that other people in the lab can execute those experiments and get trained as a scientist as well. And for my stage of my career, I'm still considered an early investigator. So I still get into the lab and do experiments myself. I write a lot of grants, so you can't do experiments without money. So I'm trying to obtain money for what I'm doing, writing our results up as a publication for scientific journals. Pre-COVID, a lot of travel can come with this. So you can go to different institutions to give talks or go to conferences to learn from each other. Now, most of this is virtual given the pandemic, but once we have a vaccine, actually this will swing back up again. And then there's a lot of service to the university that we do as well. So I thought it's kind of fun to think about how my trajectory evolved over time. So full disclosure, when I was 6 years old, I wanted to be a waitress because I idolized my grandmother and that was what she did. So she was a waitress. But I was very interested in science at an early age, and probably around that age is when I really became interested in science at school, and it's where I excelled. And so when I was 16, the natural thought was, well, I'm going to go to school and I'm going to be an MD and I'm going to have patients and I'm going to be a doctor that is in a clinic and helps patients. I wasn't sure which type of doctor. And then it shifted to a pharmacist a couple of years later because I became interested in that career trajectory. But then when I went to college, I started working in a research lab, as a lot of our undergraduates do here. And that's really where my interest in science really came to life, and I started doing a lot of lab work. And I said, oh, well, there's other things you can do other than being an MD if you're interested in science. And so around the age of 21, I've decided, okay, I'm going to go and train and go to graduate school. So I applied to graduate school. I successfully got into Johns Hopkins, as was already mentioned, and finished my training in 2010. So this is a picture of my colleague and I. And actually what's really cool is Greg and I are faculty members here at Pitt together. So it came full circle. We were in graduate school together, and now we're together as faculty members. But after you get your PhD, that's not the end of it. You refine your research skills through a fellowship, and then hopefully attain a faculty position if that's what you want to do. So I've been at the University of Pittsburgh for about 5 years now. And this is just some vignettes from my mentorship journey. Every step of my mentorship journey has yielded some sort of great advice. So as an undergrad at Vanderbilt, I learned from my first mentor to be persistent and build a foundation of learning as a scientist. And actually my persistence is what got me my first job in the lab. I kept going back because I really wanted to work for him. And he finally took me into the lab, and it was really great. And that's where I got most of my human tumor immunology, or my human immunology training, which is what my niche is now. My graduate mentor, who is still a mentor to me to this day, was an MD PhD. He really taught me the value of failed experiments and what we can learn from failing, which you do a lot when you're in graduate school, I'm not gonna lie. So it's a learning experience. My postdoctoral fellowship, that post-training that I mentioned in Colorado, I was in Denver. So Jill really taught me how to write, and she taught me how to communicate my science effectively and think about presenting my science to different audiences, which I still do to this day and I'm quite passionate about. And then it doesn't end when you become a faculty member. It's an ongoing learning process. So here at Pitt, Dario Vignali is a key mentor of mine. I have several others, but he really has taken me under his wing and has helped me become a better scientist. Before I move into the material for the webinar piece, I just wanted to also mention that you don't have to give up your personal life if you want to be a scientist. I think this was something that was told to me when I was, training, that it was going to be very difficult to have a personal life and have a family. But I can tell you that I've also done that since I've been at Pitt. Actually, I met my husband, his name's Rich, when I was 29, when I was still in Colorado. We moved to Pittsburgh together. We had our first child at 34, and now actually this is a little outdated because my daughter's almost 3. She'll be 3 in January. I did probably have children and get married later in life. That was my personal choice. I was very focused on my career, but there's never a great time. So if that's something you also want, you don't have to sacrifice that to be successful. You can still do both. Okay, so on to the topic for today. So the first thing I just want to mention is what does this spread look like when we have a viral transmission? And so this is a very simple picture of if you have somebody that's infected. So if we use COVID-19 or we use the flu or whatever you like as a virus, as an example, transmission spreads very quickly. We know now how most viruses spread, and particularly we've learned a lot about COVID-19 over the last few months, given the pandemic, but it basically can spiral out of control quite quickly. You can see this branching that, one person infects 2, 2 infects 4, et cetera, and it continues on. And with COVID-19, I think one thing that's been really emphasized is that if this is the time since infection, and this is the number of cases, if you have an uncontrolled transmission, which means if you're not taking any sort of precautions, this is what the curve looks like. And in order to do what they say in the news, which is flatten the curve and go to this blue line, we really want to do some of the recommendations that have been brought upon us, such as wearing masks, especially for an more aggressive strain of virus like coronavirus, washing hands frequently, social distancing, etc. So, when COVID started, people made it seem like washing your hands was a new thing. It's not. We're told to do this all the time. And that helps bring down the cases and helps with the healthcare system that is often tapped. And we saw that in some areas that they were struggling with the number of new cases. However, the immune system also can help with this, and a vaccine can help with this as well. And so I bring this to your attention because we're doing some of these things, but obviously bringing the immune system in and bringing a vaccine in will also be advantageous. Oh, so before I go on, Edwina, can you initiate the first poll question? So you'll see a poll question come up. I'd like you, before I go through, just the first one, please. What does the immune system do? You can answer both of them since they're here together. So there's two questions. What does the immune system do? And you can select more than one. So I just want to understand what this group knows. And then also whether or not a vaccine can stimulate an immune response, true or false. So we'll give everybody about 30 seconds to answer. And Edwina, since you're in control, you'll see when you have most of the answers, and then we can show the poll questions. You're muted too, just so you know.
Can you see the poll questions? Yes.
Yep.
Great. Okay, another 5 seconds. Okay.
Okay, and you should be able to, it should give an option to share.
Oh.
Great, perfect, beautiful.
So someone in the chat did ask, and I wonder if this is a typo, although I did copy it. Is it simulates an immune response or stimulates an immune response?
It can be either. Either word would work here, but
Okay.
I meant simulate. But yeah, either is fine. Okay, so great. So let's review them together. So basically, the number one answer for the first poll question, recognize and eliminate foreign invaders, that's one that most people got, and it is exactly what your immune system does. And the two that I expected and anticipated to have less votes, but are also both correct answers, so it was a trick question, all three were correct, is the other beauty of the immune system is that it remembers foreign invaders. And so it basically remembers these pathogens, and it helps us make sure that we have a secondary response. And it helps us get specific responses to these invaders so that we're targeting the right cells and getting rid of the right infected cells. And then most of you got that it simulates or stimulates an immune response. Either one is correct, and that's why vaccines are so important. So I'm going to go ahead and close this. Great, nice job. So, so if we go through quickly, then what the immune response does, and let me just make sure I can, yep. So it does protect from attack from foreign predators or foreign invaders every day. And these are some examples here. In the middle, you see a viral particle, but it can protect against bacteria, parasites, etc. So it does effectively eliminate all of these pathogens. To give you an idea of where your immune response is harbored, I already talked about a tonsil, but also your lymph nodes and your lymphatics are quite important. Your spleen, those are secondary lymphoid organs, but there's also certain areas such as your small intestine that have hubs of immunity as well. So these are areas in which your immune response reside, and there's a direct interaction between your lymphatics and your blood system so that your immune cells can get to where they need to go appropriately. So here's a curve showing you, it's a very basic curve, but it shows you what happens with memory and why the immune system is important for memory. So the y-axis is showing the amount of antibody that is made by one of your immune cells called a B cell, and that goes up when you have a primary immune response. And those are specific for whatever particle that you're trying to make a response to. So in this case, if we're talking about COVID, it takes some time for this to develop. This is called adaptive immunity. But the beauty of this is that when you get a secondary exposure to antigen, you get a larger immune response and a faster immune response. And so what happens is this can occur naturally, this primary immune response. You could just get the flu and then you shouldn't get the flu again that season because you've seen all the viral particles unless it's a different strain, which can happen. And I'll talk about that here in a minute. But most of the time you'll be protected the second time around. This can also be accomplished with a vaccine. So we can prime your immune response to have seen that particle or see that pathogen so that when you see it again, you don't get symptoms and you don't get sick. And so one example I can give you that wasn't occurring when I was a kid, but happens now is you get vaccines for the chickenpox. So you basically get a vaccine so that you don't get the chickenpox, and it's reduced the amount of cases that we see in children because they get vaccinated at an early age. This is an infographic, a very simple infographic of those players in the adaptive immune system. So showing basically a germ or what we would call a pathogen or a virus that is taken up by cells. So macrophages and also dendritic cells take these cells up and they basically eat them, and then they can educate T cells. There's two types of T cells, a helper T cell that basically can help educate other cells like B cells to make copies of itself to then make antibodies to target that germ or that pathogen. And then a cytotoxic T cell is a killer cell, so it can directly go and kill that virally infected cell. So these are all the players that are getting revved up during this primary immune response to basically have a good immune response, to have specificity, and to target that viral pathogen, and then to also remember it down the road. So this is why a vaccine for COVID is so important, because if we could prime our immune system early on to see this virus and to see what's going on, we would have less individuals getting sick, and we would also then have some memory associated with it. Now, there are some caveats that we've seen in the media with COVID-19. I won't get into them today, but there's a lot of worry about the secondary response not being as robust. It's possible that that could be the case, but memory cells will be there even if the antibodies are not there up front. And so it's just a matter of reinvigorating this memory response properly and learning how to do that with these patients. This is another more detailed snapshot of the innate versus the adaptive immune system over time. I won't go through this for the sake of time, but it's more of a reference for you. So you can see your first line of defense, and skin is really your first line of defense to a lot of pathogens. And your innate immune system kicks in within hours. And it takes several days for this adaptive immunity to kick in that I was just talking about. But this is really where your specificity and your memory is going to come from with in regard to a vaccine, or if you've seen a pathogen before. Okay, so talking about why we should get vaccinated, I thought I would use the flu as an example, since we don't currently have a COVID vaccine that's being implemented. And as I mentioned, Andrea will talk about that and where we're at. So these are the number of cases that are projected for the flu, for the common flu this year. And this is what is going to happen as if individuals get vaccinated. So you can see the amount of reduction in hospitalizations and deaths and the number of cases within the United States as a result of getting a vaccine. And so that's why we say you should go get vaccinated every year, because it will protect you as long as it's not a different strain that you come into contact with. And that leads me to my next slide, which is why is it difficult to make flu vaccines, or why it might be difficult to make a viral vaccine like for COVID. And so basically these viruses are quite complicated. In the case of flu, if you think about it having 3 types, you have different types that can be more dangerous than the other. There's different proteins that you can vaccinate against in order to get that immunity. And sometimes it's not clear which proteins we should be vaccinating against. Sometimes we don't hit all the right proteins, and so they might get missed. And so that's why some people still get sick despite being vaccinated, but yet they're developing that immunity naturally, so that's okay. And also viruses can change quite quickly. And so, viruses like the flu virus or COVID can mutate and change. This is called antigenic shift. So therefore, there could be changes that occur well after the vaccine's already manufactured that could affect the population downstream. So that's some of the complications that come with making a vaccine that's effective and potent for most of the population. Lastly, I just want to mention herd immunity. And this is quite an important thing as well, because what happens is if you don't have individuals getting vaccinated, you see this spread of infection. But if you have people that are getting infected naturally and developing immune response or getting vaccinated, those are these individuals in green. And so therefore you're protecting the population because you have more people that are immune. And so this is basically the definition of herd immunity. It's basically preventing the spread of infection because there's pre-existing immunity and a high proportion of individuals as a result of either natural infection or vaccination. So that's what we're aiming for and what will help us and what helps us every year with the flu as well. Lastly, I just want to end on talking about why some of this is challenging in COVID patients. So here's that picture I showed you earlier showing some of the key immune cells that I mentioned. So T cells or CD8, B cells or CD20 that are present in a human tonsil or in a secondary lymphoid organ. This is a normal individual. This is somebody that has not had COVID. So there's been multiple studies that have come out that have shown that if you look at individuals that don't have COVID here on the top and patients that do have COVID on the bottom, you can see those that have COVID, these germinal centers are these sites where education occurs and the immune response is revved up, are dysregulated, and they're not as intact as they are in a normal individual. So this could be a result of the infection is something that's different than flu, for example. And so therefore that immunity might not be getting built up as well as we would like it to. And so in addition to a vaccine, there's a lot of other therapeutics that we're thinking about that actually work well in cancer patients to amplify and reorganize these structures to then get better immunity in the long run. So with that, I'll end and I'll save questions for the question and answer part so that I can shift to letting Andrea share his screen, but I hope this was a good background for what's happening with the immune response. So let me stop sharing. Oops. Oh,
thank you, Dr. Bruno.
Yep. There we go.
If you do have a question, you are welcome to put it in the chat and then we'll address it shortly, just so you don't forget your question.
You're muted, Andrea.
Can you hear me?
Yeah. Yes, thank you.
Thank you, Tullia, to set me the stage to present today some of the work that we have been doing in the last 20 years in the field of vaccine development, in particular in developing vaccines that are effective to coronaviruses. So I will give just a brief background of my particular background. So I grew up in Italy. I was born there in Bari, which is here in the southeast part of Italy. And I went to medical school there where I got my degree in 94. Right after my degree, I started the residency and during my residency in 95, I was interested in gene therapy, in genetic engineering. And I had some background in genetics, molecular genetics. So Pittsburgh was a hot place to come and learn gene therapy. So that's what I did. My initial plan was to come 6 months, but here I am 25 years later, still here in Pittsburgh, working on basically the same things that I came from, genetic engineering, for different purpose. So one of my main tool in the past 25 years has been this virus. It's called adenovirus. And since 95, I learned and I developed this as a technology that is a technology that is commonly used now, but back then in 95 was not a well-known technology. So we contribute in develop this is an efficient genetic engineering technology that can be used for both vaccine or infectious disease vaccine or cancer treatment, so immunotherapy approaches. So what we do with this virus, which is a virus, the strain that we use is a strain that causes a common cold in human, adenovirus serotype 5. We cut and paste the genome of this virus, attenuate this virus, and cut the pieces of other viruses or bacteria, and make hybrid viruses which the immune system recognizes as something different. And we can make the immune system recognize this virus as an influenza virus, as a coronavirus, an HPV virus, and any virus that we want. Sometimes the immune system will buy that, generate an immune response that can cross-react with those other viruses. That's what I was doing in 2002. These were the target vaccine that we were working back then in 2002. Our main work was back then in HIV, but we set the base. We learned this technology, how to develop this vaccine in this virus, which is called Epstein-Barr virus, which is the virus that causes mononucleosis. And we were using different technology, and adenovirus was our main technology. We were using naked DNA. Back then, the technology was called gene gun, and also recombinant influenza virus vaccines. And the targets were HIV, BB, influenza. There was anthrax, was a bacteria that was emerging in the early 2000s. There was also a veterinary disease that I was interested in, is a sheep disease. It was a blue tongue disease. This was 2002. Toward the end of 2002, there was this new emerging respiratory syndromes that emerging in southern part of China, in the Guangzhou city, in the Guangdong province. This was discovered to be a new coronavirus. So we had the coronavirus back then. We had a human coronavirus type 1 was the coronavirus 229E, and the OC43, the coronavirus type 2, which were causing mild respiratory infection. About 30% of common cold are caused by this strain, a mild strain of coronavirus. This was different because the pathogenesis of this virus was completely different from this mild coronavirus. Was really causing severe acute respiratory disorders, causing that the 10 to 15% of people infected were dying of this disease. So me and my group start to study this virus and see how we could tackle this virus. So we tried to see what were the weak spot of this virus. This is the structure of a coronavirus. We usually target a protein on the surface. And the main protein, the prevalent protein on the surface of this coronavirus was and is now the spike protein. The spike protein that the coronavirus is used to enter. So we decided certainly this would be a very good target, but we choose also to see whether we could tackle the membrane protein or the nucleoprotein. Again, this was the beginning of 2003, where we built the first 3 coronavirus vaccines. These were based on adenoviruses encoding for the N protein, nucleoprotein, the spike protein, and the membrane protein. And we tested this vaccine in mice and monkey, and these are the monkey neutralization data, showing that this vaccine will, especially the spike encoding vaccine, was inducing neutralizing immunity against the coronavirus infection, but also that the nucleoprotein was inducing more T cell. It's another harm of the immune response. So we reported this study was December 2003 in the Journal of Lancet. This was the first coronavirus vaccine that was developed in the world. And we set the stage for the spike protein to become the main target of coronavirus vaccine future development. We had an effective vaccine. We manufactured a vaccine. We were hoping to test this vaccine in 2004, the beginning of 2004. But suddenly this disease, which never reached the pandemic stage as the SARS-2 did this year, disappeared in 2004. As the disease disappeared, also the funding for the disease disappeared, and all opportunity that we had to test this in a clinical study basically disappeared because there were no money, and so we decided to move on other target disease. We work on several targets, mostly influenza, HIV, Ebola, until 2014. 2014, there was the same event happened. There was a new emerging respiratory viruses that emerged. This time emerged in the Middle East, in Saudi Arabia, and concurrently also in Qatar, which is still they are both in the Middle East. And this virus was similar to the SARS, was a beta coronavirus, was using a different receptor though, and this virus was named MERS coronavirus. Now, while the first coronavirus intermediate host was this animal, this is a civet cat, which in the animal market of Guangzhou infected the first patient. The first patient was actually exactly 18 years ago, was was November 17, 2002, that the first reported patient. So this was happening 2012, which yes, was 10 years after the first patient of SARS. So this was the intermediate host. This time was a camel, dromedary camel, and this is usually all these beta coronaviruses also beta coronaviruses are bats, and bats then you choose an intermediate host, which is could be for the SARS-1 was the civet cat, and for the SARS-2 is the dromedary camel. Then for the MERS, the dromedary camel transmitted the virus efficiently to human, but we never established an efficient human to human transmission of the MERS coronavirus. Thanks God, because despite that the MERS coronavirus is still circulating mostly in Saudi Arabia, the efficiency of transmission from person to person is very low. And this is good because the mortality of MERS coronavirus is roughly 35% of mortality, while the SARS-1 coronavirus mortality is around 10%. So this will be devastating if a MERS coronavirus make the jump, the molecular jump, the molecular mutation that makes it more transmissible human to human. And this is a virus that will keep working on, because we fear that this could be potentially causing pandemic. So we did develop the first vaccine for MERS. It was in 2014. We reported again in a publication in 2014. And we show again that we're using this technology with adenovirus and using the spike protein. These were different formats of spike protein. I'm not going to go in this detail. That was an effective mean of inducing a response in animal models, and and we continued to work on the MERS. We had some funding, and we tried to develop the second generation coronavirus vaccine. That was back in 2016 when I partnered with Dr. Lou Fellow in the Department of Dermatology, which he was working on a cool technology. And this technology was a cool way to deliver the vaccine. The delivery is based on the patches. These are microneedle patches. These are microneedle of sugar, in which we can put the vaccine inside, and this vaccine is released in the skin once this patch is applied to the skin. So in 2006, we designed alternative vaccine. This time we're a subunit vaccine. These are just piece of protein, again, protein of the spike fragment of protein. And we studied we compare the delivery of the patches with the delivery with just regular injection. We found that when we were delivering the vaccine with patches, the immune system was giving a much stronger immune response, even if we were using much less vaccine. And we stick with this vaccine, and we work for until we were working in 2000 until the end of 2019. We were actually developing a vaccine for camel, because the rationale was if we can immunize camel, we can protect basically the host of this Middle East respiratory syndrome, so we can block the transmission to human. It is an indirect way to protect the human from being infected. And here is the study that we did. This is 1 year on 53 weeks. We show that even after 53 weeks, the immune response after this was after prime and boost immunization is still very strong at week 55. So it's a persistent. And this happened that, while we were manufacturing, we have a contract with Saudi Arabia for the camel vaccine. The SARS-CoV-2 appeared in January. Actually, in December last year, we were following this new emerging infectious disease until in January was reported to be another coronavirus, very similar to the SARS-1. This time, the intermediate also was this interesting animal that is called a pangolin. Again, the primary host is the bat, and the bat should choose the intermediate host, which is the pangolin. and the pangolin transmitted this, possibly this virus for the first time in September, October of 2019 in the animal market in Wuhan in the southern part of China again. And it started the pandemic, and so we decided back here in January 29, when the study, when the virus was reported, the sequence was reported, we were able to generate a vaccine in only 12 days from that sequence became available. So 12 days later, we have a protein, recombinant protein, that we stick in our microneedle array that I showed you before, these little patches, and we use it to immunize mice. And very quickly, we generated in early March, we had already the preliminary data showing that, again, that this SARS coronavirus was susceptible to the induction of antibody against this spike protein. And this spike protein was a protein that was neutralizing the viral infection. So we're still working on developing this vaccine. And so these were the preliminary data that we generate for that publication showing the induction of immunity. And we started the manufacturing of this vaccine as a clinical product to start clinical trials to put into the patient. This is just the characterization of a clinical product. This is a fancy molded top that showed the high pure vaccine. These are different state of the subunit protein, as a monomer, as a trimer, as in this natural form. And today we have tested 26 different vaccine prototype. We down selected now 2 products. This the Pittsburgh, which is hopefully in the end of December, we should be able to start the clinical trial. This is under review at FDA now. This is our patch vaccine that we hope by the end of December, in the beginning of January, to start the clinical study. We are very confident that this vaccine is really potent vaccine when we compare with other technology that we have. We have also an adenovirus technology, adenovirus vaccine, which encodes for spike and nucleoprotein. This is for both humoral and cellular response, but we are advancing in parallel as a clinical product. And so these are just a cartoon to show that this is the clinical study that we are starting in next month here in Pittsburgh. Is a prime boost study where the vaccine is administered to the patch. We have generated already the GMP, the quality vaccine. We tested, we released, and we have a very good preliminary data in the animal. These are some of the moment of our production. This is our GMP manufacturing facility. We make some of this. Now we go do small scale, but good enough to test this vaccine in a human clinical study. This is when we vial this vaccine that goes then goes in the microneedle array. And this is the second vaccine that we are developing. We are testing at the cancer center where Tullia work. And this is the adenovirus. The difference is this vaccine is given as a intranasal spray. This is an atomizer, so it's atomized, and we plan this as this vaccine as a single immunization. Again, this vaccine is very potent in inducing both humoral and cellular immunity. And this is just a quick manufacturing. So the way we manufacture first a stock of virus, we call master viral bank, which we bank. And then with this bank, we manufacture the drug substance that then becomes the final product. And the final product is eventually is the one that we use to manufacture. These are the doses that we prepare for this adenovirus-based vaccine. These are 750 doses that are waiting since July to be administered, and we're waiting for the final approval from FDA. I stop here, and I'll take any question if there are any. So this is briefly our experience in developing this particular SARS-CoV-2 vaccine that date back almost 20 years now.
Thank you very much, Dr. Gambotto. We have quite a few questions in the chat. If you wouldn't mind just stop sharing your screen, that would be awesome. Thank you. And can you see the chat, Dr. Bruno and Dr. Gambotto?
Oh, not yet. Let me see.
Yeah, I can see it. I could probably just I can read them out, and then Andrea and I can decide who's the best person to, you know, take it, or we can do it together, basically.
Okay. So there's a couple on immunity and some about vaccine development and effectiveness.
Yeah, we can just start. We can start from we have time, I think, to cover most of them. So,
okay.
So the first question is do we know if the SARS-CoV-2 virus is changing a lot such that it may affect the vaccine development? And so this would be a good one for Andrea to take.
Yeah, so the virus is changing, but so far the changes that are in this protein that is important for make the virus escape the immune system, which is the spike protein, those changes don't seem to be good enough to change the vaccine product. Actually, we tested our vaccine was designed on the Chinese initial strain. Now there is a prevalent European/American mutant. It's called the DS14G mutant, which is the prevalent. Actually, the vaccine that we have, the Chinese vaccine, works much better in neutralizing the new version. So the new virus is attenuating itself. And this is it's very common in virus. So virus try to adapt. So a good virus doesn't kill its host. So if the virus kills its host, like MERS, SARS, Ebola, it doesn't go very far. And SARS-2 is doing a very good job in not killing enough. It's killing a lot, but not enough to just self-limit itself. And these mutations are going toward that direction, which is a good news for us, because we don't need to run and make a new vaccine now. So we can just keep working on what we are doing as and all other 150 vaccines that are out there. So we still are good with the product that we have now.
Okay.
So the next question is on testing for the presence of memory cells for COVID-19. So there's a lot of ways to test for immune memory, but basically if we think about B cells and T cells that are the main producers of memory to the virus, there are actually canonical kind of markers we use in immunology to mark those subsets. So we can basically use a technique called flow cytometry to quantify what's present in these patients in their peripheral blood or in their nasal passages if we get tracheal aspirates. So for our study that we're doing locally, Dario Vignali and I are leading a study looking at the immune response mainly systemically in these patients. One observation that's come out is we've started to cross-compare patients that are discharged versus patients that are cease to breathe or pass away. And so basically in the patients that are discharged, they have many more memory B cells, and we see them pop out of the analyses that we're doing, and they're very pronounced. So we can measure them by this immunological tool to understand how many are there. You can do a lot of other tests as well. Like you can ask if it recognizes the virus by doing some other immunological techniques, but you can also just survey the population of cells that's in there by drawing blood from an individual and seeing if memory exists. One question is about halting research and how that's halted research for other viruses, and if so, how big of an impact will that have on future research? I can just say that like a lot of the initiatives at the National Institutes of Health, that is a lot of the funding source for a lot of us as investigators, has shifted its focus to trying to encourage investigators to get funding to study COVID-19. However, we haven't really seen a direct impact on, like, for example, funds to study cancer necessarily. They are not getting shifted, but Andrea can probably talk about other viruses and what that's doing.
Yeah, I think that's that is true. So all the interest that is shifted on the coronavirus. but we advanced the field of vaccine development in the last 10 months. In the last 10 months, we did basically progress which would have taken probably 20 years to make if COVID-19 would not appear. And this is the ability to quickly develop a vaccine and test it. So we learn how to quickly, how to design a trial to make sure that the vaccine is effective in a very short period of time. If you consider that this virus appeared 10 months ago and we have already two vaccines that are ready to be licensed, that is incredible. And that technology and our technology, whatever we are learning, can be translated on other viruses which are don't have enough funding, enough attention, which are in fact remote part of Africa. I mean, we can apply this technology, the patches, the RNA technology, to all other targeted. It's been an incredible experience, an incredible journey and advancement. I think this was, at the end of the day, was a positive aspect scientifically, and it will advance the field of vaccinology for sure.
And then the two questions that are kind of next, they go together, I think, because one is about concerns on long-term health impacts from the vaccine, since it's a new vaccine method only tested for a short time. And are there vaccines, example of vaccines where there are long-term side effects that weren't seen until years later?
So there is no vaccine today that has shown a side effect 2-3 years after. The only main vaccine that show some important side effect was the respiratory syncytial virus, RSV vaccine in the 60s, 70s was developed. It was actually, the vaccine was potentiating the infections. And but for some of disease, you can see also from the epidemiology for the infection itself, like the dengue, that you can have an enhancement effect. The preliminary testing for SARS-CoV-2 and also the pathogenesis, the clinical history of this disease for the first year that now that we know patient we are pretty confident that is not going to be the case. It's unlikely that we're going to see long-term or enhancement type of effect for a vaccine. So I'm pretty confident that this is going to be a very safe vaccine. It's relatively simple to tackle.
And I can take the next one, which is asking about why there might be differences in how people respond, such as severe disease versus asymptomatic, and could it be due to the immune response? The short answer is yes, that does play a role. Particularly when this first occurred, we were seeing that the elderly community was being impacted more severely. And that's due to the fact that as you age, and I didn't get the time to talk about this, but as you age, your immune system also ages. So the responses that you once had when you were younger are not as robust as you grow older. And so we definitely have individuals that go into a group called immune fragile, which means that they don't have as robust of an immune response. But that's probably only one piece of the picture, quite frankly. Like, there's other factors. And I showed you just a snapshot of what could be happening in individuals within their lymphoid organs that could be affecting their immune response. So that could have something to do with it. But I think that the immune response does have a lot to do with that impact and that response. The other thing that somebody asked was differences due to ACE2 receptors. So there's been a lot in the news about ACE2 receptors and how those cells get infected. What I can say about that is that we've done a pretty thorough analysis via that technique that I mentioned, which is called flow cytometry, where we can look at individual cells from a patient and look at the expression of different proteins on the cell surface. And so we looked at some of these different molecules that seem to help the virus enter, such as ACE2. Also, neuropilin-1 is another one that's been in the literature. And we see differential expression of those molecules on certain immune subsets. So it actually looks like there's certain immune cells that are more prone to being affected. And it's definitely different in patients that do better versus do worse. And so we're still trying to understand that expression. But Andrea, I don't know if you want to add anything to that.
Yeah, as you said, there is differential expression. There are different organs that represent the expression of ACE2 receptor, And these are the organs that are usually involved in the pathogenesis of SARS coronavirus. So it goes from the heart, kidney, brain, and these are, of course, lung. And you see the infection. Of course, to have infection of the virus, you need this receptor. But one important thing is the metabolic aspect of ACE2 receptor, because the fact that the virus uses a lot of this receptor kind of sequester this receptor to its natural function, which is converting the angiotensin. Angiotensin, there are different forms of angiotensin. Once the angiotensin, there is one angiotensin that induces vasoconstriction, and one angiotensin that induces vasodilation. And ACE2 converts one into the other. And if you block the receptor, then you have only vasoconstriction, and that can cause vascular problem like a thromboembolism, which is a frequent effect that is seen in those patients. So the receptor is very important for the pathogenesis. In fact, the MERS has a relatively different it's worse, but no, it's different the clinical picture because it has a completely different receptor. So they don't use the ACE2. ACE2 is used by SARS-1, but not by the MERS.
So Edwina, do you want us to keep going, or do you want us to kind of wrap because we have a couple of minutes?
To kind of wrap it up, there are a couple of questions related to vaccine development that it would be nice if you could just comment briefly, maybe just a couple of minutes. So one person asked about the process of disclosure of information on new vaccines. Where can you find information? Is it, will the CDC publish information about the new vaccines, for example, Pfizer? Another one is how accessible will vaccines be? And then also, Pitt is developing their vaccine. Is that vaccine competing with Pfizer and Moderna that we're hearing about in the news? And so what will happen? What will be the outcome with that?
Let me elaborate in general, give you a general response. So we are developing this product, and so the microneedle vaccine this will be the first microneedle vaccine to be developed. But we can set the ground to a novel technology. The advantage of microneedle is easy. Everybody can put it itself. So you don't need any special tool. You just push this through your skin. It goes inside, and after about 10 minutes, it's completely dissolved. You take the patches out, and you're done. So far with the testing that we have done for this and other vaccine, we have shown that the vaccine is stable. So it's stable at room temperature, so it can be shipped in the mail. You can UPS this vaccine all over the world. So this is a huge advantage. There is no needle, so needle are important things in the vaccinology. So you want to try to avoid needle, because a needle can be a problem in underdeveloped countries. And go on. So the vaccine itself, the format, it doesn't need anything else. The vaccine is the actual product is a needle in which you have the formulated vaccine. So there are several advantages. Of course, if you are successful with this vaccine, this can set the stage for the new influenza vaccine. All vaccine will look like this, and they are more accepted as a vaccine. So my kids will like better my patch than a needle, especially my daughter, that she doesn't like very much the needle stick, like many people. But no, that will be a big advantage. And then this vaccine, the dose sparing that will happen with this vaccine, because the way we put the vaccine. So microneedle array will be a big advantage, a big advancement in the vaccinology field. There is competition. There is not really competition. At this stage, no, we are all tired. We all want to get over with this disease. We're not competing. Probably nobody's going to become millionaire with this vaccine, at least not me. And the information are, as soon as one of these vaccines become available and effective, nowadays the news will do their part to the CDC, but all over the world will spread. So even data that are not complete, that are not official, are now in one day they become worldwide. So if our vaccine or other vaccine become available or are effective, we will know. In the long term, this vaccine is a success for the science. The advancement of any of these vaccine will be really a success, because these are all new products, all new things that can be used for other diseases, and hopefully by middle end of next year, we will be all vaccinated with one of or the other product. And very likely it's going to be the combination of those products, because none of this vaccine can satisfy the 6 billion dose and that we need to immunize the worldwide population. 6 billion dose, that means primary boost is 12 billion dose of vaccine. Now, there is no product that has ever been produced in this quantity.
Great.
Well, that sounds quite thorough for answering. Dr. Bruno, did you want to say anything before we kind of wrap up?
No, I think this has been great. We covered a lot of the key points, and just to say that there's really insightful questions, and I think brought a lot of discussion to the table, which is what we were hoping. So it's great.
Well, thank you very, very much. We appreciate your time and effort. Participants, thank you for joining us. And again, this video will be processed and eventually posted on YWIB online if you want to watch it again or share it with your friends and family. So best of luck. Everyone stay safe, and thank you very, very much.
Thank you.
Thank you very much.